Awesome. We're going to go ahead and get started because we have a pretty packed agenda and a lot of exciting presentations. My name is Renee Julian. I'm a librarian actually at Florida State University, and I'm chairing this panel where we're going to talk about persistent identifiers and fair data, fair instruments. But first, I want to introduce our sponsor. Brian Leland is going to be coming and talk from Oxford Nanopore, is going to be joining us to talk about his company. Brian Leland is a bioinformatics manager in the commercial applications group at Oxford Nanopore, where he works with research hospitals, core facilities, and other external collaborators. Prior to ONT, he worked on the applications team at Inscripta, which is a CRISPR startup, and he trained as a wet lab scientist at NIH and Yale, where he studied mechanisms of DNA repair. So please join me in welcoming Brian, sorry.
All right, thank you for that introduction. Thanks to the organizers. So today can you guys hear me in the back okay? All right. So today I'll be talking about the analysis tools we have for nanopore data, epitome, and how they can be used by anyone at any level of expertise. At Oxford Nanopore, our mission is to enable the analysis of anything, anywhere, by anyone. To do that, of course, we have a set of kits and sequencers, but what we're focusing on today is analyzing the data. You can generate really rich and complex data, but if you don't have a way to analyze it, it's not going to be useful to you. So that's what I'm going to talk about today. There's lots of challenges in analyzing sequencing data. Can be time consuming, you have to learn new tools, really poorly maintained tools sometimes. And so what we have is these set of solutions to reduce the amount of time, make it easy for anyone to do, either via point and click or with a command line, and give you confidence in your analyses. I think the clicker is not working for me here a little bit. It's weird that it doesn't show me what's happening on here. Oh.
It's not advancing.
It's not advancing.
Yeah, but it is here.
It's advancing there, but not there. Oh.
All right, well, I'll be the guinea pig for you guys before.
A lot of times you have to press this. And oh, here you go.
Here we go.
Okay, which one did you want to go back to?
It's still different there. Interesting. Now the arrows are working.
Is this where you?
Yeah, that's good. Okay. So we have a whole suite of analysis tools that we've developed at Nanopore for all different kinds of applications. Along the bottom here, I'm going to focus on the two in the middle, epitome, which can be used via our desktop software or via the command line for bioinformatics teams. So epitome, what can it do? So it enables the analysis of all types of data for different applications, from metagenomics through germline, human genomics, tumor normal samples, direct RNA sequencing, and single cell. We previously had a cloud version, but now in 2024, we're consolidating everything into one desktop application, Epitome Desktop. It's really easy to use and yet very powerful. And most importantly, it's free. So as a core lab, this is really excellent because it's free for you to use. It's free for your users to use. If they have collaborators, it's free for them to download and use. So it's very accessible in that way. How does it work? So it's a standalone desktop application. It's really easy to install and use, and it's compatible with Windows, Linux, Mac OS. And what it does under the hood is it actually runs these full end-to-end bioinformatic workflows built in Nextflow. So in this way, using the GUI, the desktop application, is giving you the same rich, deep analysis that you can also do via the command line with these pipelines. The pipelines that it packages do all the standard analysis that you would want to do for that application, and they use a bunch of different tools developed by ONT and community developed tools, and they're all vetted and strung together in an intuitive way, end to end, so you get the full analysis from sample to answer. And so, taking this really complex workflow, it saves you a lot of time because all of this is linked together and you can start the entire run with a click of a button. What do you get out of these reports? So when you run it, you get really nice interactive reports that help walk you through the kind of top level metrics of your data. But you also get the raw output files that are really richly annotated so that you can dig in and extract a lot of interesting detail out of your experiment. So what I'm showing here is an experiment that I worked on with an external collaborator. It was a human whole genome sequencing. We get all of the variant calls, about 5.3 million, which is a lot to sift through, but because of the rich annotation provided by the pipeline, we can dig into only the variants that we're interested in for this project, see how each of those impacts different genes, and ultimately look at only the likely deleterious variants so that we have a smaller set to look through as we move on to tertiary analysis subsequent to this workflow. You can also integrate your own workflows with Epitome. So the desktop application can run community developed or pipelines that are developed by other bioinformaticians that you collaborate with so that those workflows also become more accessible and easy to run through the desktop application. And all of the workflows, like I mentioned before, that power Epitome desktop are Nextflow workflows that are written and highly tested by our bioinformatics team. And you can run those same workflows via the command line, and they're all open source. So you can go and see exactly all the steps that are being run by the pipeline at every stage. So if you want to come learn more, there's lots of links up here, but you can also, of course, stop by our booth, booth 213, and I'll be available here after the talk if anybody's interested in long-read sequencing and long-read sequence analysis. I'm happy to answer any questions after. Thanks.
Hey, look at that. Hi, everybody. My name is Renane, again, and I am here to talk about our project, which is an NSF-funded project called Enabling Open Science Through Persistent Identifiers for Research Instruments and Facilities. And it's a research coordination network, and it's a 3-year project that we started last year. So we're in year 2 of our project right now. This is a collaboration between 4 different institutions. So we have Florida State University, we have the National Center for Atmospheric Research, we have the University of Colorado Boulder, and we Stanford University. And we have a mix of folks that work in core facilities, librarians and scientists as part of our project team. So the main goals of our project are to create a coordination network, an RCN, that's focused on the assignment of PIDs specifically for research facilities and scientific instruments. And so what we want to do is collect use cases over our 3 years for why and how PIDs can be assigned for facilities and instruments. And we want to, during this process, we want to facilitate the generation of expertise and guidance on key topics, get the right folks in the room to start these conversations. And then towards the end, we want to provide recommendations and lessons learned from our work with the community so that we have guidelines and things like that. So we're going to start with a couple working definitions because they're going to help inform the other 2 talks too. And so PID is short for persistent identifier, and they're just essentially a globally unique machine resolvable link that's associated with the metadata scheme. I'm not going to read all of these definitions, but I listed a couple examples below. An instrument, essentially, the definition we're using is a device for making measurements, and then obviously facility is a little more straightforward. But just take a quick look at those, just so as we move through facilities, instruments, PIDs, RIDs, and things like that, you all have an idea of what we're talking about, even though you probably already know a lot more about this than I do. So we want to encourage, wait, hold on a second, let me show them on the right side. There we go. We want to encourage the use of PIDs, mainly to increase transparency in science, reproducibility in research, equity in science, and that's why a lot of the folks that were part of this project team were really interested in applying for this grant a few years ago. You know, this is an example of sort of network science and the thing we're all sort of working towards with assignments of PIDs and direct object identifiers, citing data sets, things of that nature. The idea of creating a network of science where not only we can track the impact of publications, but also data sets, instruments, core facilities, and everything else. So I'm going to show you, there are a couple examples at our local institutions, and a lot of this is actually work that Claudius has led at different places that he's been. And so the first example here is, this is Stanford, right? Yeah. That's Claudius's work with Stanford applying RRIDs to certain instrumentation there. This is work that's happening at NCAR, which is actually at UC Boulder, or CU Boulder, where they are assigning PIDs for specific instruments that are listed here. And then, so we wrapped up the first year of our project in December. And what we did is we built a website, which I have linked here and also at the end of the slide. But we spent a lot of time talking to people. We were on the phone a lot. We touched base with our awesome advisory board. Some of them are here in this room and on the panel. We went to a lot of conferences. So I was here last year for the first time ever. And we did a poster on the idea of our project and some of the things we were going to try to do over the next year. And then we did focus groups and presentations. We had focus groups at FSU at the Highfield Magnetic Laboratory. We had focus groups of folks that worked at core facilities at Boulder. And we just sort of were trying to generate low-hanging fruit and figure out what some of the barriers of PID adoption for facilities and instruments were. And then all of our work kind of ended with a workshop at Boulder, which the main deliverables, one of the primary deliverables of our RCN is to actually have workshops that are in person where we bring folks, you know, all in the same space and we talk about the challenges and where we can work together to sort of increase adoption. And we did our first workshop, and that's what I'm about to talk about. So we had 35 folks that represented, you know, many different states. We had different disciplines from biomedical, environmental, space science, materials. And the way the workshop worked is it was a combination of presentations, similar to the ones you're about to see and hear in a minute. And we talked about examples of PID adoption, things that were going on at the local level, and then we would break out into focus groups. And the members of the project team, the PIs of the project team, would lead focus groups, which are subsets of our workshop participants, and we would ask them questions about, you know, do you use the PIDs? What's your familiarity with systems? If you have used PIDs, what are some of the challenges to assigning them to instruments? And we also just published a workshop report that summarized our results of not only the workshop, but the first year of our work as a project team. But I'm going to spend the last couple of minutes of my presentation going over our primary observations from our first year of work in the workshop. And the first is that there's a need for this. You know, there's you know, encourages reproducibility, data provenance, things like that that I mentioned earlier. And so we, you know, we were hoping that that would be an observation, but obviously we wanted to list that to just in case it wasn't obvious. And this next one is kind of obvious too for many of you who are familiar or trying to become more familiar in this area, is that there's different systems and different tools in use. And so it's scattered and it's kind of inconsistent on which PIDs are being used. Sometimes multiple PID systems are being used to document, you know, facilities or instruments, but really the idea isn't at this point to pick one, but to really just like make people aware of all the ones that are that we're finding as we go through. And we're actually working on a PID graph as a project team now during our calls. And we hope when we have it basically looks like a pretty Excel spreadsheet with different systems and what they are supposed to do and links to them and things like that. The next is adoption. So I think I kind of spoiler alerted this, right? It's less about picking the right tool and more about making folks aware of all the tools that do exist and making sure that people working on things know about the work that their other colleagues are doing. And so we want to develop guidance on navigating the systems. Like I mentioned, the PID graph that we're working on now is a step in that direction, and it's really helping us understand the landscape. And next is metadata, and as a librarian, I found this one fun. We need to think about that. It's one of the biggest things we found in our focus groups is there are so many things that need to be captured for a specific instrument, and a specific instrument can change as you add parts or remove parts or make changes to it. And so, you know, that was a big thing that we are looking at is the work that other people are doing in trying to create a schema. So there's a group called PIDs. I don't think that's how you say it. But these are folks at DataCite that are actually working on a metadata schema recommendation. Oh, RDA, sorry, thank you. Yeah, RDA, not DataCite. That are working on recommendations. And so we're working with them and trying to just keep our ear to the ground and see what they're working on. I'm kind of bleeding into these as I go through the recommendations, but granularity, I mentioned that a little bit. What exactly is a scientific instrument? That is a question that came up a lot in our focus groups. And so that is something we're going to need to talk about as a group. You know, right now the idea is about adoption, so it's really more important to capture PIDs for as many instruments as possible. But as we do that, we're going to run into an issue of the same instrument could have multiple PIDs once you make significant modifications to it. So how do you track the versions over time, and how do you have a unique identifier for the right one? The last few are a little more straightforward. The resource limitations, right? This takes a lot of work. And so we want to gather the challenges that the community is facing in PID adoption so that we can pass that information along to folks like publishers and funders and things like that. And then the next is convincing people of the value of citing PIDs. And so this is kind of pushing the community. This is the, like the broader scientific community, PIs, facility users, and really just, you know, I think people who operate a facility understand the value in their facility being cited and tracked, like the research being tracked by a citation in a paper or including the PID somewhere. But that might not be as inherent to folks using the facility. And so communicating the value and the importance to all stakeholders is going to be something that we're going to want to do in the next couple of years. And really just thinking about incentives and how they may increase PID adoption is something that our group wants to think about, because there is a need for incentivizing not only the creation of PIDs, but also the inclusion of PIDs in papers and citations and things like that. So very quickly, this is our project website. The slides will all be here, but you can check out our website by scanning the QR code. And our next workshop, the second one, is actually going to be in Tallahassee, Florida, and it's going to be in a few months, and we're going to be emailing folks in the community, making you aware of this, and we hope that to see a few of you there. All right. Let me make sure we got the right one. That's okay. Yeah. Here you go. Let me set you up real fast. There you go.
Okay, so hi everybody, and thank you Claudius and Rene to invite me to present the work that actually I've been doing with Anita and Jim, who is here, and others. And this is also, in addition to the people that you see here, there is a lot more people behind all of those nice little logos, which one of which is some of which you know, RRAD, Core, Marketplace, and ABRF, but then there are other ones that I encourage you to learn about if you don't know about, because we're all trying to work together to ultimately improve data management and sharing. And in particular here, I'm going to talk to you about, I'm going to give you an introduction about the use of PIDs for this purpose, for more of the user perspective, and then Anita is going to tell you more the technical aspects of a lot of this. Oh, maybe I have to do this. How do I advance?
Right arrow should be good. Let's see what we got going on here.
This is the talk of technical difficulties.
Yeah. Point. I don't know
May
why.
have to restart the PowerPoint presentation.
Really?
Last time that had to happen.
Really?
I can also
Okay.
advance maybe here. Do this. Yeah. No, that doesn't work.
No, I think you have to close it
Yeah,
and
you have to close it and reopen it.
Like actually close it, quit. Okay.
close it and reopen it.
Escape.
No, push the other button.
Control delete testament.
I'm trying to do quit.
You could do an interpretive dance.
Sorry, I'm not, I don't use PCs normally. That one.
Are you just trying to click over to the next slide?
Yeah, but it always works with you, but it doesn't work. So good.
Remember Renee
Yes.
earlier? You want to come over here?
Sure.
Okay.
So,
You have to click the, was it Brian or which one of you guys I
Yeah.
showed? Yeah, you always have to click this so that you can be able to right click.
all right.
Okay?
So, Anita,
I now know. I
maybe we'll, okay. All right, so, so here I'm going to tell you a little bit, I'm going to take a step back, so why all of this is important, what is needed, and how are we going to do it. And, you know, PIDs is one of the pieces, is not the whole thing, but it's, we obviously here are convinced it's one of the important pieces. So research data management is following the whole life cycle of data, and it starts from planning and then actually about then sharing and reusing your data and other people's data. So there is an aspect that has to happen every day so that you organize your data, and this is directed more at the data producer, part of ourselves, because often we are both data producers and data users, but let's put on the hat of the data producer. We often are, it's really important for us to keep track of the data we are producing, and for facilities, we are really very much involved in producing data, often for other people, or helping other people produce their own data. So that's really important for facilities. And then there is something that happens after a project is concluded, at least for now, and it's being published, and we want to make available that data to further use. So there is a pre-publication and a post-publication data management, and so one part is more about management and organization, the other part is more about sharing, but management and organization happen in both sides. So, because even during data production, we want to share with colleagues, lab members, ourselves, our future self, and so on. So it's like overall, we could call it day-to-day management and then sharing, but these categories are not necessarily, you know, it's just a way of starting to think about it. So why this is important? Well, we all know about this, the lost data map. And this is about us producing our own data and actually not finding it. Or somebody, a colleague has left and we're not finding it. So this thing is actually talking about ourselves, a gift to ourselves in the future. We want to have something so that we actually find the data. And one thing that I want to point in this, you know, image, obviously is very different from, you know, what we are going to actually end up using for biological data. But it's about boxes. Each one is separate, and there is the label. Without a label, they might be very organized, but I'm not going to find them. And then, of course, if they're not in boxes, but, you know, you could also think of a dump as a storage, but obviously it's not going to be very useful. So storage is one part, but organization and labeling it's an essential part. And, you know, why is it important? Quality and rigor, reproducibility, sharing value. And it's all, I don't think, I'm not going to spend too much time addressing all of these aspects that are all very important and many of us are aware. And then, of course, there is the big carrot or stick at the end. In other words, NIH is actually now requesting for us to do sharing, management and sharing, to post what is our plan and then enact this plan. There, you know, at least in the policy, it's written that funding will be subject to success, you know, the actual implementation of this plan. So something we need to talk about. And in this context, I'm sure all of you know, but I want to remind you about what FAIR is findable, accessible, interoperable, and reusable. So we want to make, and all of this is about future-proofing your data. But it's not just future for like posterity or for another person and being a good citizen. It's primarily about finding it for yourself and organize it for yourself, and so that you can get more impact from your research. So what is needed? So this is just a few of the things. Data formats, so we need to have data that is in open formats so that we actually can read the data that other people produce. We can continue reading the data we produce using maybe software that, you know, that now that we have lost the license to a certain specific proprietary format that we don't have access anymore. Metadata is about actually making sure that we document the data with rich information that is machine readable and can be attached to the data and moved along in the data pipeline lifecycle. And then software infrastructure, this is where, for example, PIDs come in. You know, so in order for us to use PIDs, first of all, the PIDs have to be, somebody has to take the responsibility to creating them and managing them, and then we can use them. So there is a whole lot of stuff that has to happen that we are not necessarily directly responsible for, but that is really important. And I want to stress the idea, the term infrastructure. We all think about infrastructure in terms of physical things. Here, you know, I think we need to start thinking about infrastructure in terms of software and what is the whole organization of under the hood stuff that ideally, if this is done well, we don't have to worry too much about it, but it's nice that it's there. It's like a car engine. Most of us, at least me, don't want to know anything about the engine, but I want to be sure that the car moves. And I'm glad that there are people that actually takes care of the engine for me. Because I cannot do the fun things with the car without that. So open formats is one of the first thing I told you about. So it facilitates the data exchange and analysis. So it facilitates keeping track of the documentation, exchanging the data and the analysis so that we can give to the people that will do the analysis, if they're a colleague in our lab or somebody else in another lab, the ability to access the data in a way that is easy for them to then develop the readers. Actually, they may not have to develop the readers anymore because they are available because the formats are open. There are multiple different examples here. Some of them you may be already familiar with, but you know, they have to do with multiple different data types, and some effort is in many fields to generate open formats. Then there is metadata that has to be rich but organized also. And again, this concept of a gift to your future self, and then also is a gift to others, which is also good. So to be useful, it has to be easy to track and retrieve. And now I'm going to give you an example. Actually, these are the wrong slides. Okay, no. So, no, it's not the wrong slide. Actually, it's good. So the first thing is I'm going to give you an example for microscopy. So this is actually similar to the slide that our commercial partner was presenting. There is sample preparation, image acquisition, image data analysis. And we have to keep track of all this information. And all of this information collectively can be called image metadata. And it has these three categories that pertain to the three phases. But this applies to everything else. So there is there is experimental and sample metadata, instrument metadata, analysis, and visualization metadata. This is often, pretty much every kind of different data production process has these phases. And typically there is a huge fragmentation. These information are all over the places. Very difficult to keep track of them, very difficult to keep them together with the data. So we have a data, and then we have this information, and they're often all over the place. So how do we, for example, could do it? One way of doing it is using spreadsheets, and especially this is very important for experimental and sample metadata. For instrument metadata, often we can get the information, sometimes if there is good, well done formats and standards, we can get it from actually the headers of the files. But a lot of times we may need to augment that with specialized tools, such in this case, this tool that my lab developed in microscopy to collect the metadata for instrument description that is not provided by the manufacturers. And then for the analysis, it's about keeping track of all the steps. It's hard to do for the analysis, but guidelines are being released in different fields. And in this specific case, this is a guideline that was released for the imaging field. Regarding experimental metadata, there are different ways in which we can keep track of the metadata. One way is to put some information on the file, but then file name, but then that becomes really huge. Another way is to organize your data in subdirectories. Another way is to actually have information in spreadsheets that can be saved as CSV file or other formats that are easy to share. So the idea of using spreadsheets it's like it's a good starting point for sure. All of us are using them anyway. The point is that because they are very powerful, but the question is how do we make them actually more future-proof? Because they are often difficult to interpret, even by ourselves and definitely by people that might need to analyze our data. So how do we make them more future-proof? So how can we organize these spreadsheets so that they are actually more easy to, more valuable, let's say, for us and for others? So one way is to start thinking about using shared vocabulary. So for example, we can talk with other people in the core or in the lab about what are they doing. Can we start agreeing on some common terminology vocabularies? So if you share the terminology and the language, the structure of the Excel spreadsheet in your lab is going to be easier. And if you actually go past your lab, it's even better. Use the idea of using ontologies. So these are lists of shared terminologies that have been developed that can be used instead of just reinventing the wheel and use new terms. And then, of course, metadata recommendations and guidelines are being developed in multiple fields that can help us doing this. Once we have created a little bit more of an organized template with some shared language, what about sharing it with other people? And maybe even before we start producing our own, we can see if other people have already produced similar things that we can reuse. Then there are also more advanced tools that are spreadsheets that use ontologies that allow you to more easily enrich the spreadsheets with ontologies. There are different tools that I'm listing here that are out there and that are being developed. They're more or less easy to use. They tend to be relatively easy to use, so this is something I encourage you to look into. And then, of course, the idea is that at the end, the metadata has to be captured in formats that is easy to to transfer, so XML, JSON, JSON-LD, but just even CSV, it's another way of doing it, or Excel spreadsheet. So, and then another important aspect is the embedding in all of this information of persistent identifiers. What's happening? Okay. One thing that I want to bring you to your attention is that there is this group, one of the organizations that I'm also part of is Bioimaging North America. There is this group that we have started and is meeting every 2 months, which is called, it's written there, Automated Image Management and Metadata Annotation User Group. It's a user group, so the idea is that we're meeting every 2 months to share different experiences that people have, tools that people are developed. It's obviously centered on imaging in this case because it's Bioimaging North America, but if anybody is doing imaging and may want to just start seeing what is out there, what people are developing, this might be, it's a very open place where to meet. It's on Zoom, and you can go to the BINA website to register. So now I'm going to talk about the persistent identifiers. So you already heard about it, but I just want to give you just a refresher. There are multiple, first of all, there are multiple ones, and one of the issues that also was mentioned before is which one is appropriate for what resource. And, and the idea that we don't want to have too many different identifiers. And one of the questions that people often ask is which one am I supposed to use? So here it's a little bit of a guide of what I have learned so far. So there is ROR for institution, ORCID is for, most people know it's for people, RID for core facility tools and reagents, PID inst for instruments, and then there is DOI, which many people know for data and publications. And definitely in the adoption part, we will have to figure this part out, which to use for what, because it is very confusing. But one aspect that, one of the points that was raised before was metadata is important. Think about the PIDs also as a place where a metadata can be associated with an entity, so that some of the metadata can be with the PID, and so you don't have to put all that metadata, for example, in your Excel spreadsheet because it's already available in a place where it can be retrieved easily, and you don't have to then use the, keep track of the metadata yourself because somebody else is keeping track of that. So there are, the idea is that, we can then start putting things together and link these things together. So for example, if you are a core facility, you can have a list of the instruments that are at your core facility, but now with persistent identifiers, it's much easier to actually, the added value is that now you can actually have a link to a specific one instrument and some of the characteristics of the instrument. Then the PIDs, then you can link to the instrument the data that was produced and the paper that was used, that came out of that research. So that all of this stuff can be connected, and then you can imagine that this network can become really big. An example for microscopy was something that we have, is what I'm going to conclude with, is the work that we've been doing with Anita and Jim and other people to think about how can this be done for a microscopy instrument. And the idea here is that, there are all these different sources of metadata, and often they are all over the place. So PIDs could help us keep these various aspects, for example, the description of the instrument, associated with the image data. And then the image data is associated with the sample description and other aspects. So PID can help maintain these links. So what we did was we brought together one side, which was the core marketplace effort that was started by ABRF and the Vermont Bioresearch Network, and had a major input from USDIT, which is actually a project that Claudius spearheaded. And then with FTI Lab and SciCrunch and the RRAD effort, with effort that happened, with other efforts that happened that I'm more involved in, which is Bioimaging North America and QUAREP-LIMI. And this is an organization that includes a lot of different bioimaging scientists with instrument manufacturers to develop quality control and reproducibility. And so basically, we brought together this team to develop a plan that we call imaging PhD, so imaging persistent hardware descriptors that allow to actually tag each instrument with the PID and then associate with it persistent descriptors of this instrument so that all of these different other information can be kept linked to each other. So the core facility can have an inventory. They can use this inventory for budgeting and funding. They can be put in experimental protocols and scientific publication associated with the data and with the repositories where the data then is going to end up going. So I'm done. Questions? Or maybe we have first, yeah.
Okay. All righty. Thank you, Caterina and Julian. And thank you all for sticking around. And that's, why does that say AACR? That's not the right talk. What was that?
I can find your slides.
Could you find my slides? It would be really cool if they weren't from AACR. I can tell you about them.
Actually, weren't they up here earlier? Weren't they?
were.
They
No,
were?
I thought they were.
Yeah, so I don't know where they went. Yeah, okay.
80%? Could you find my slides?
Like your slides were right here and now they're not on the thumb drive. They were right
Well,
there.
they're not on the thumb drive. I
Right.
think you moved them over, you moved over the AACR ones.
What do you mean moved over?
Oh, I don't know.
This is you guys, right?
I think I need to reload her slides onto, can I just grab that thumb drive?
What's your name?
Anita
probably need that thumb
Bandrowski,
drive.
Bandrowski. And that's from, see that? That's from AACR. That's a whole different meeting.
Gotcha.
My computer again. I just need that thumb drive.
Yeah, if you can grab
The
that.
piece that you gave me, Caterina?
Let me talk about publications.
I think it's,
They also have the same thing in your email.
So thinking about
the one that I don't have the connector, the connector is Anita's.
Do you want a handheld?
at some point,
Okay.
everything will be gone.
Do you want a handheld?
Oh, yeah. I'm okay. At some point, we will all be gone. We'll be dead. There will be dust, right? This starts really well. These kind of talks start amazingly well. What will be left of us? Let's think about this. Now, some of us have children, and some of... Yeah. Some of us will have a little PowerPoint adapter. All right. There's this thing. Here.
I don't know what happened.
Okay.
We're going to test it.
Right. Back to death. So, one of the things that I would argue we as a society have agreed upon is that we will maintain the literature. Better or worse, we will maintain the literature. If you were to see a slide here, there would be a lovely quote from a long dead Greek. And the quote would say something to the extent of, man cannot step into the same river twice because it's a different man and it's a different river, right? But why do we know that? Well, the reason we know that is because we have something that does not move like a river. We have the scientific literature, and we have the literature more broadly. We have agreed to maintain the scientific literature. It is what we count when we say, how many papers have you put out, right? That's an important thing, and everybody can argue about how right that is, how wrong that is, and impact factors suck, and they're wonderful, and all of that stuff. But at the end of the day, this is the artifact that we will maintain. This is the artifact that will outlive us. So, my argument is, let's take a look at the literature to figure out, hey, how are we being represented in that artifact that gets to stay around long after we're dead, hopefully? Unless it's retracted, in which case bad things happen, right? That looks really good, thank you. Can I get click-throughs? Oh, can you, oh, there we go. Click. Does it click? It doesn't? Yes, it does. Oh, look at that, see, there's a guy, right? Okay. So, going back to our river, right? And Heracles, I can't pronounce his name. I'm just going to tell you that. Thank you. Thank you. Fantastic guy. Has some books. Right. So what are persistent identifiers, right? If we understand that a river is something you cannot step into twice, it's the same sort of thing as the internet. Believe it or not, the internet changes all the time. You search for something, the next day you search for something, and the result is different. Why is it different? Well, it's like a river, right? So what are persistent identifiers? And how do they relate to the scientific literature? Because when we write papers, we want to reference things that are out in the internet, right? But that internet is a river. So persistent identifiers with proper metadata are like little rafts. They hook up to the things in the river that we want to talk about, and they stabilize those little bits. And then we can take those bits and stick it into that final form. And so I want to tell you about how we do that. So just to get everybody's attention for 5 seconds, this is one of those persistent documents. And this document says, hey, here is how we're referring to this particular reagent or resource. It is a mouse anti-flag antibody from Sigma. And one might say, oh, that's perfectly fine. That's a great way to talk about a reagent. Except the problem is that when you go to Sigma, you actually get a bunch of them. And when you come back to Sigma the next year, I did this talk once with a single antibody for about 4 years. And every time I would change and actually do that search in Sigma to see how many antibodies there were, and it went up and down. But which ones were there when the person ordered it before? So that is what happened. So let's look at how cores and instruments are actually acknowledged. Here's a lovely way to acknowledge a core. Now we know from the core director that in fact this is the citation of this director's core. Now that Keith person is the former core director 2 directors ago, by the way, and that was never the name of the core, right? And that's not the right number, but it's close. It's close. So it took some time for this core director to actually get back the, whoa, that's really pretty, get back the information about this acknowledgement. It took him a long time. And when you use actually the name of the core or core PI name or former PI or some staff member of the core, the less reliable that information becomes, the less other people can actually find that information. Things that are more reliable are these RRIDs that we had heard a lot about. Those are pretty reliable. They have a set of metadata that's even machine actionable. Grant numbers are more reliable. URLs are more reliable, although we all know about URLs and how about 25% of them undergo something called link rot. That is when that river moves on, and also when somebody in administration, nobody here from administration, right? Okay, I'm going to talk bad about them right now. All right, anybody from administration gets to decide that, hey, this is great to redo the entire university website, and it destroys all of the links and all of that interlinking that you all have done for, what, 15 years? And then everything else is, yeah, has to be redone. Okay, well, you know, things happen. So the less time you have, the more you would like to find these papers that actually reference the RID, the URL, or other fun things like that. All right, so to show you what a tool page looks like, this is a core facility. This core facility is called Duramab. It's recently been discontinued, but it's a core at UC Davis that does a lot of antibody work. And what they have is a bunch of basic metadata. They have some relationships to other core components, to the institution. They have a bunch of usage metrics, because we can text mine things like, hey, did you use a URL for this? Did somebody use an RRID for this? So we find that information. There are even ratings and alerts. And if it is a core, it also gets registered with the core marketplace. And so, as Caterina had talked about, there's been a project and an ongoing effort to try and make sure that when you enter your information into the core marketplace, or if you enter your information about your core into the RRID project, that we share that information so you don't have to do it twice. We try really hard to not make you do it twice. But of course, RRIDs are not just for cores, and they're not just for instruments. They are for all kinds of things that National Institute of Health has listed a bunch of things that they really care about, which change from lab to lab especially, and they are the reason why experiments go bad. Reason why experiments go bad? Antibodies. Cell lines. Cell lines are not authentic. A good chunk of the time, we want to make sure to at least name and shame all of those specific resources, right? Because if we can name them, at least we can figure out information around them. And that's going to be one of my asks for you guys. Okay, so do authors actually use reasonable information when asked and when these identifiers are available? Sure they do. They will do what they need to do to publish in Cell and or any other journal that they really want to publish in. And so here's a nice, lovely Zeiss microscope with its RRID, and you know what? That link, that was put in by the journal. Those links can be put in by the journal to your core. Kind of fun. How do authors do this? Well, they go through, you know, they go in and basically the journal tells them, go to this website. They go and search for their things, including your core, then they copy and paste that little piece of information. So this is really important, guys. If you have a core and you have a website for your core, these are all good things, and then someone tries to cite you, right? They're trying to cite you. So they look up in your website where it says, here's how to cite us. If you write a diatribe, a 5-paragraph essay, right, on how to cite you, they're going to move on, right? They're going to have something else to do before reading that 5-paragraph essay. The core marketplace gives you this little snippet of text, the name of your core and the RRID. And you can just say, cite me like this, copy and paste that little tiny snippet of information into the paper. And if you do that, the authors can actually do it. Fabulous. So, if you need to register a new core facility, magical things have happened on our website. You can go right to the core marketplace from the register a new type of RID. There's no need to be logged in for any searches on our site, and also no need to be logged in at this point. But if you type in, if you click on the core facility thing there, you'll get right to the ABRF core marketplace. And then, of course, which journals are doing this, and I'm sorry that my slides are so bad looking up here. I swear they were better. But Cell Press actually has a lovely table that lists every single reagent that the paper had used and every resource. Cores usually don't go into that, but software tools do. A lot of instruments have also been popping up in this list as well. How do you know that we've made any difference, any impact at all in anything? Well, this graph. Here we text mined the heck out of the scientific literature, looking for every single antibody that we could find in the open access literature. We found over 300,000 of those mentions, and back in the 90s, 10% of them were associated with a catalog number. But if you look right there in 2014, when we started the RRID initiative with 25 journals, we're starting to change the slope of that blue line a little bit. And that blue line really shifts when big journals like Cell and eLife actually come in, because they are much closer to requiring the RRIDs than anybody else, and it sort of shifts that entire curve up. Journals like OncoTarget, which never did a darn thing with us, are coming along for the ride, which is great. Okay, so what is the way that you can actually get your stuff cited? So as I said, if you have a 5-paragraph essay, not going to work. But if you have something that's really easy, and this is the website for one of our cores here from the core marketplace, there's a little cite this facility. This echoes exactly your RRID for your core. So if you're registered with Core Marketplace, you already have this thing. Use it. This is our latest project. We started to ask the question, what is impact of these little persistent identifiers? Is there anything that we're actually doing to improve the scientific literature? And this is what we found. So what is that graph? This is a set of over 1,000 papers that we went through manually. We looked at every single citation of every single mouse put together by this core facility. This is one of the animal cores. And in 2011, half of the papers named the animal using a nickname. Using a nickname. And the way we know that it's a nickname is because the name is short. And when you put it into their website to figure out what it is, you get 3 mice or 15. We don't know how many you get, but not one, right? You don't come up with one. And so if you look at that orange bar, see how it changes? As soon as we get into the RRID project, there aren't that many mice that are being referred to by the RRID. When the core actually puts on their website, this is how you cite the mice. That's in 2017. It's at the end of 2016 when they started to do that, so we started to see the papers. In 2017, you see that big bump in the dark bars at the bottom. Doesn't go up to 100%, but it does go up a lot. It went up to about 25–30% almost overnight. And then it keeps going up, not as fast as we'd like, of course, but it keeps going up. But the other papers are using catalog numbers. So more people are using catalog numbers, and the category that we're getting rid of is that nickname category. which means the scientific literature goes from 50-50, this paper is irreproducible because this mouse is not findable, to 90-10. That's huge. That could be you. All right. So we looked also at the cores themselves. Here's a couple of the same kind of an analysis on two different cores. They're both microscopy cores because of Caterina. And these two cores, one of them you can see that's been around a really long time, and one of the reasons, so it got an RRID right around the same time as this other newer core got an RRID. But see, the difference between those two cores is this is the old dog, new trick, right? And the other one is new dog, new trick. And what you will find there is that a lot of these people either didn't cite the core properly. The reason that we know about this is because those lovely, lovely, hardworking people that are in this core facility went through the scientific literature and they associated each paper with a grant number. Every once in a while, authors will also do this, but largely that's work done by the core facility itself. But after a couple of years, we're getting to be about 20% or so of the papers that have an RRID associated, which is great. It's not where we'd like to be, but it's great. That's a little bit easier to associate. With the newer cores, what you'll actually see is after a couple of years, they got their RRID in 2021. There was a single paper in 2021. And then in 2022 and 2023, a really good chunk of their total papers actually had RRIDs. So that's great because they're teaching their users how to cite them, and their users are actually able to do that, right? And then we can easily find what that means. And this is a fun part. I just wanted to bring to you your attention the fact that, in fact, research resources can be good, and if any of you worked with antibodies, they can also be bad. And antibodies are wonderful, but the problem is that sometimes they're not so wonderful. So cell lines are another one. This is a paper that my student, Juliana Babic, actually put together. And as you probably very well know, some cell lines are contaminated. They are problematic. They have issues. And the question that she asked is, if somebody knows about those issues before they publish the paper, even if that RRID mention, right, which is right here, this is the web page that somebody would search. They would do their searching, and they would copy and paste that RRID. But right down there, what it says is problematic cell line contaminated. This one's been shown to be a HeLa derivative, right? That's a problem. Do authors see that, or do they ignore it? The answer is in that graph. They see it and do not use the cell lines by decrease of 66% over a text mine version of the scientific literature with 150,000 cell line mentions. We looked deeply into those 50 papers that actually had used a problematic cell line, and they were all good to use except for one. One person out of all of these papers ignored that kind of a warning. Everyone else, these are benign things. Things like HepG2, which is actually a problematic cell line because it was misidentified initially. Hepatoblastoma, hepatocarcinoma. What are you going to do? Well, don't use it for the wrong disease. Yeah, besides that. All right. So we actually have created tools to assess the quality of research, which is very much outside of the impact factor. And we are starting to see when cores, like the animal cores, really push their users to improve how they talk about not just the animals, but all of the things around those animals, that it's starting to work. So we have this metric. It's maybe not the best metric, but it is a metric that's very much orthogonal to the impact factor, to look at the quality of reporting in those papers, and we're starting to get a significant change. We're starting to see a significant shift, but only for papers where the core facility is actually pushing their users to do a better job. So again, this is going to be my ask to you guys. Can we help rigor and reproducibility? When you write the methods section parts that I know you guys are writing for these papers, can you make sure that this is done? Because you will impact a much larger size of a literature compared to, a regular lab. We know that these numbers actually do mean something. This is from the Cancer Reproducibility Project. We ran these numbers on top of the original papers that we know were not reproducible from the cancer. This is the Center for Open Science version of the Cancer Reproducibility Project, which everything was done in the absolute open. And you can see that the replication studies are far more reproducible. They're kind of centered on 8 out of 10 as opposed to 3 out of 10. So 3 out of 10 looks like it's not very reproducible, but 8 out of 10 seems to be much better. Okay, so this is going to be my big ask right here. Please join us. Come help us, right? It'll help you to get your course cited better. And it'll help us. If you know things about antibodies, about software tools, I'm sure that this tool or this set of tools that was presented at the beginning of this talk is great, right? But if you find problems with it, we have a place that you can tell us about that. If you find a problem with an antibody, we have a place that you can tell us about that, and then we will put it on those little warnings at the bottom, just like cell lines. If we already know how to record these kinds of information about cell lines, and if you give it to us, we will push it to the right authority. But if you know that from antibodies, we're the authority. Please tell us. There's a way to report that data on every single RRID page that there was a problem with this resource. This is what happened. We will require that you give us some kind of data information about what it is that you're telling us. I mean, it can't be just like, my resource is better than your resource, right? But what we'd love to kind of get you guys engaged around is the quality of these reagents, the quality of these resources, because these are going to be the things that get left after we're all dead and gone. So with that, I would like to thank you. And hopefully the next talk is not going to be about death. Have a nice night.
Questions?
Hi, I'm Andy from Northwestern. I have a question about when we're asking PIs to cite an instrument, does the core then automatically get cited, or do we need to advise both?
I think you should advise both.
Okay.
You should definitely advise both. And if it's for one of these two beautiful people over here.
It's for you, actually. So can you talk a little bit about how the need for cores to include grant citations in the acknowledgements versus RRIDs? Both.
Both are good.
And
Both are good.
so you'd recommend both?
I mean, if you can get people to do grant citations, and if that's the way that you've been working with your group, that's great. We will not find them generally, but you can find them. So they're recoverable, right? The information is recoverable as long as there is a PID in there.
Okay. Thank you.
A question about instrument RRID.
Yep.
Should those not be instrument-specific serial number rather than a generic term?
That would,
So this is the project that we have done to extend the capability of the core marketplace for instruments with also Anita, and has been exactly that, at least for microscopy. So the idea of associating so the RRID will remain, it's basically the PID for the model, so that will remain that way, and so if that is sufficient, that will be used, they continue to be used, but if you want to have a PID for the actual serial number of the instrument, so for that one specific instrument, that is where the idea of a persistent hardware descriptor comes from. So this is, we submitted a project proposal to the NSF. The use case is for microscopy, but the way we have described it is that this is not just for imaging. And we are leveraging the PID initiative from RDA, the Research Alliance, that actually goes in that direction. So, and to have a minimal metadata schema associated with the actual identifier, and then extended metadata descriptor. And the idea is to issue DOIs for these descriptors.
yeah. So how many instrument manufacturers in the room? Several, okay. This is absolutely essential, right, to have persistent identifiers for your instruments. Now, I can't tell you how many times our curators have gone in to try and find a hardware description and it's not there. So, you know, all of these beautiful shiny machines that are, you know, a couple doors down, they're lovely, but tomorrow they could be gone. And I would really, really, really strongly encourage whatever you guys do on the instrument side to make sure that there are at least descriptors of those instruments somewhere on a raft, right? On that raft in that river. Because once those web pages go down and you've moved on to the next version, it's actually really hard to recover that information. So I would say, please have those available. I mean, there's beautiful librarians out there, right? They will give you DOIs. If you don't want to fill out the entire metadata PID inst schema, you know, with all of the associated stuff, those are really important, but at least have that stuff recoverable.
So as a follow-up to that, rather than simply the serial number of the instrument, so I'm an electron microscopy co-director. If someone cares to give me the money, I upgrade the camera periodically, and therefore it is a subset of the instrument that would need to be individually identified.
Yep. Yeah, so on that front, we're talking about light microscope in this case, because that's what the people had most expertise in. The idea is that there is a PID for what we decided to propose in this specific project was serial number for the instrument, but then the hardware descriptor contains information about each of the components. Up to this point, we didn't get to recommend having unique identifiers, PIDs, for each of the components, but that is definitely somewhere where one could go. But at least because the other thing that happens is that LSM 900, for example, model, often is not enough to describe actually what are the components of that specific one instrument that was sold, because the configuration can be very different. So the idea is to associate specific PID to that specific serial number, but also to the descriptor.
So I don't know if there's anyone here from the local, from Minneapolis Imaging, but at our microscopy conference last year, Mark Sanders introduced, I think it was a Python script that would write your materials and methods from the metadata.
Yep. Yeah, that's methods J2.
Right.
Yeah.
Great idea, I thought.
Fantastic idea, as long as it has a PID at the end of it.
Right, exactly. And I think you've identified, you know, the RRID of, say, a faulty cell line.
Exactly.
That could be automatically picked up as part of the code.
Yep.
If you've used this cell line, please don't write the paper.
Exactly. But really, the granularity
Yeah.
piece that, you know, Caterina brought up earlier, this is really, really important. If we want to think about this across different kinds of resources, it's the same kind of thing. RRIDs will get you to a certain level of granularity that seem to be what authors are able to give us. Because we've asked for, you know, lot numbers for antibodies. Trust me, we've asked. We don't get them. But we need to keep asking. So thank you all. I think.
Yeah, that's it.
I'm sorry we're over time. I blame the